Welcome to Inside the Hive.tv podcast, the show that takes you into the world of bees. I'm Dr. Umberto Boncristiani. One of the biggest problems in beekeeping today is the ectoparasitic mite Varroa destructor. As any beekeeper on the planet today knows, this mite feeds on the fat bodies and hemolymph of honeybees with severe consequences for the health of honeybees. Likely the biggest problem with the mite infestation is the different viruses the mite transmits. The immune system of the honeybees was never adapted for this new situation. Therefore, any solution attempting to fight virus infection is in great demand. Today, I speak with Dr. Vicent Ricigliano, a honeybee researcher at the USDA Honeybee Research Laboratory in Baton Rouge, whose recent publication describes a potential new solution against honeybee virus infections. Before we jump into the conversation, I would like to thank the team of Vita Bee Health. Vita Bee Health is the only company dedicated to bee health and is always bringing you solutions to help bees and beekeepers around the world. Thanks Vita for sponsoring this channel. Please check them out, link in the description of this video. And now, I introduce you, Dr. Vicent Ricigliano. Vincent, in the name of Inside the Hive.tv community, I want to say, first of all, thank you for your time to join us today to discuss your latest publication. And also, I want to thank the USDA for letting you to join us today and to present that, to have this fruitful conversation. Before I start this conversation, Vincent, I want to ask you a little bit about yourself. How did you get into the bees? How you get into USDA? Where you're from, your background, scientific background, things like that. Can you give a little overview about your background?
Absolutely. Well, first of all, thanks for having me. Been checking out some of your podcasts. I think it's really good. Nice way to communicate to the stakeholders, beekeepers, and you have some good production of this podcast as well.
Thank you.
You're welcome. So let's see some background on me. I was born and raised in New York, and the path to bee research, I'd say, has been nothing short of serendipitous. I've been fascinated by the natural world for as long as I could remember, so I suppose there was an interest in biology at a young age. When I got to college, I thought I would go to medical school, and at the time, I was passionate about human health, and I still am. But after getting an introduction to molecular biology or the study of genes and DNA, and also getting some experience working in a molecular biology laboratory, I knew that research was the thing for me. That's what I wanted to do, and I wanted to run my own lab someday. So I went to graduate school, got a PhD in molecular biology from St. John's University in Queens, New York. There, I studied plant and microbial biotechnology. Specifically, how can plants be harnessed to produce drug-like chemicals? And this research involved plant and bacteria, plant and virus interactions to manipulate pathways responsible for producing biologically active compounds in plants. Also worked on a project involving flower symmetry, and we were trying to understand how do plant genes influence the shape of flowers? And it's an important question because it could help us understand, well, how plants attract pollinators. And so this was really my first introduction to plant-pollinator interactions. When I first graduated, I knew I wanted to do applied research that can contribute positively to society, not just do research for the sake of knowing things, but to do research that could directly contribute to society, maybe support a healthier future for humans and the environment. And the mission of the USDA, and specifically the Agricultural Research Service, was in line with these goals. I saw a job posting to study the honeybee microbiome at the USDA Bee Lab in Tucson, Arizona. And it's hard to describe, but in this moment, I couldn't really think of a more interesting topic. I knew about the importance of honeybees as agricultural pollinators. I knew they had some serious issues, and I thought, well, maybe I could use some of this training and experience to develop new technologies to help bees. So I ended up getting this job as a postdoctoral researcher with Kirk Anderson at the USDA Bee Lab in Arizona, working on honeybee-associated microbes. I learned how to keep bees and culture bee microbes. So I got quite a bit of experience working commercial beekeeping operations. And it was there really that I fell in love with bees as an organism, but also as an agricultural system that interfaces with other agricultural systems. There's really nothing like it. And I knew this is what I should be doing. So flash forward to now, I have my own lab at the USDA Honeybee Research Unit in Baton Rouge, Louisiana. And here we are. And I could talk a little bit about my research there if you want.
Yes, but before we jump there, so I'm trying to summarize here to my own mind here. So it's fascinating sometimes. When I ask those questions to people that I interview here, I also I heard all kind of stories, and your story was kind of shaping into getting to the pollinator. You know, it's very interesting. Everything you were doing was kind of, you know, was a funnel to get there. All the little pieces of knowledge that you accumulate throughout your career bring you to there. I think is a fascinating how people get into bees, and another story for the people at home to how incredible those situations are. So yeah, can you give us a little bit, you know, an overview about the goal of your lab and the goal of the Baton Rouge lab as a whole too?
Yeah. So in general, the Baton Rouge Honeybee Breeding Genetics and Physiology Lab, they've historically been focused on breeding mite-resistant bees. And now these breeding programs have expanded for other traits of interest. I do some bee genetics related work, but I've been mainly focused on pathogens and nutrition. I could talk a little bit about that. But first of all, I have an incredible lab group. It's really a great team. I have 2 lab technicians, Victor Rainey, Daniela J., postdoc Alexander McMenamin, grad student Ellie Martin. And the main goal, I'd say, of my research program is to develop practical technologies and treatments for the beekeeping industry that can be applied to hives to alleviate major stressors. And, you know, I want to be able to stand in front of beekeepers, be able to explain what we do, and have them nod their head in agreement like, yes, that makes sense to us, and that is useful to us. So that's kind of the guiding force when developing new research projects is, you know, thinking about that first. And we focus mainly on pathogens and nutrition, as these are the 2 most modifiable determinants of bee health. So regarding pathogens, we're currently working on developing new antiviral treatments, and we're taking a few different approaches to do this. One approach relies on RNA interference, or RNAi, to stimulate the bee's antiviral immune system to target viruses. This approach is related to the paper that we'll discuss today. But the other route is practical antiviral treatments for bees using natural product drug discovery approach. And here we're screening diverse natural sources for antiviral activity and their relative toxicity to bees with the goal of developing natural treatments that could be delivered as nutritional supplements. So
that's quite interesting. So you are screening natural compounds too against the virus infections in honeybees.
We are.
Cool. There is a publication out there that I don't know or I'm not aware of, or of something you can't share yet.
Of us, yeah. So this is currently kind of undergoing, and you'll see it as kind of a dual approach or a split approach, where we're taking this kind of genetic engineering approach with organisms that interface with bees as kind of one route, and then we're taking a totally natural drug discovery route, but both aiming to improve bee resistance to viruses.
Which is one of the most important topics, right? In recent years with in the beekeeping world.
Absolutely.
With Varroa mite coming in the equation, and now potential other mites coming into the equation, the transmission of viruses and all those viruses reaching tissues that were never exposed before create an exponential problem for the bees to survive. And we're still on this going. We are not in a balance yet, so the bees don't know how to handle those new virus routes and infections and multiplications and changings. And so it's a constant battle, and technology can definitely help. That's why I was very excited when I saw your publication, because I am a big fan of RNAi. I worked with RNAi before. I was a CTO of a company that was developing RNAi solutions against citrus greening and mosquitoes, and I know the potential. And the main reason why I'm a very big fan of RNAi is the potential to reduce agrochemicals. It is in my core after visiting 10 years here in the United States, visiting commercial beekeepers and farmers. I believe people are crossing the line with the chemicals. I mean, we need to find ways to reduce the chemicals that are spread everywhere. And RNAi is target-oriented, very specific, do not stay in nature for too long. I didn't see any data to go against the approach of RNAi system. I don't know if you agree with me on that statement, if you know any data against RNAi in general.
No, I don't. I mean, there's some questions of maybe specificity and maybe off-target effects, but of course, before RNAi is ever deployed, would ever be deployed in the field, there would be thorough risk assessments to test those specific molecules before they ever saw the light of day. So no, I don't know any, and I share the same opinion about the potential of RNAi to reduce agrochemicals and increase agricultural sustainability, including beekeeping.
Yes.
So maybe leading into the paper that we'll discuss, I'll just say one more quick thing about another major focus in the lab. It's improving artificial diets. As I mentioned before, nutrition is a key modifiable determinant of bee health, and development of artificial diets is kind of lagging behind that of other livestock and companion animals. So, we feel that it's possible to produce an artificial diet that fully recapitulates the nutritional and functional qualities of pollen. It just hasn't been achieved yet. We're taking a few different approaches to this as well. But one approach is developing functional feed, or diets that influence bee health beyond just macro and micronutrients. For example, if you've been following any of the publications, our recent publications, we've been studying the effects of microalgae feed additives. And some species of microalgae are a good source of macro and micronutrients for bees. We found that chlorella, a green algae, and spirulina, a blue-green algae, also called the cyanobacterium, can improve bee nutrition and naturally stimulate bee immune functions. So we're currently characterizing these effects of algae in bees and how we could use them to develop practical treatments.
And I have a whole plan for that. So with the publications that are coming from your lab. So stay tuned. I really enjoyed that. I have that. Some of the papers are in my list to cover that, but I need more time for that. I still a one man band here. So cool. So this is fascinating. This is not only fascinating, it's very important. You're covering nutrition and pathogen, very likely the two main stressors that put beekeepers in trouble out there. No questions about it. So, Vincent, let's go and enter a little bit of jump into the article itself, the article that we're gonna discuss today. Let me put here, because I want a little overview directly from you.
Sure.
Let me put here on the screen. This is the publication that I want to talk about today. Green biomanufacturing of edible antiviral therapeutics for managed pollinators. If you need to explain this to a beekeeper in a couple phrases, like what's the main message? What have you done here? No jargons, no scientific jargons. How you explain what you got here and the importance of that?
Yeah, so I've been thinking about this concept almost since I began working on honeybees, and I started asking, what if treatments could be grown in some edible biomass like plants or algae and specifically tailored for bee health? And if this works, this could be a sustainable approach to help bees without the use of any synthetic chemicals. And it could be adapted potentially to emerging threats and pathogens. And also, the paper reports that a novel technology using something called RNA interference or RNAi, and it's a conserved pathway. It helps bees fight off viruses. And when the bee's immune system detects viral double-stranded RNA or dsRNA, its immune system is triggered to degrade those RNA sequences. And that's really the mechanism by which this technology works.
So I want to stress this because I have some problems sometimes to explain this to beekeepers. But RNAi is part of the immune system of honeybees. People might not know that, but it's a conserved pathway against virus infection. It's already very well demonstrated that RNAi is activated by double-strand RNA, which is one of the replication stages of the viruses that infect honeybees. So this is very conserved in the immune system. It is part of the immune system of honeybees, and we're just discovered these things in the last 20 years, and right now potential new products are coming to the market exploring these new discoveries from a decade or two ago. So just to be clear, RNAi is part of the immune system of honeybees. Not only honeybees, actually mammals too, and all cells, mammal cells. I don't know how fungi. I don't know how far down we have this already figured that out, but it is part of the immune system and the defense mechanisms of cells in general against virus infection. So I just want to make that clear. So what you did here, basically, if I understood, you got an algae and you make this algae produce this stimulant of the immune system of the honeybees to be targeting a very important virus of honeybees. Is that a fair message? Is that the fair way to describe the article?
Absolutely, I'd say that's a good summary. Yeah.
So where the idea come from? Why do you use this algae? Can you give a little overview?
Yeah. So like I said, I was thinking about this pretty much since I started working on honeybees. Just this concept that what if treatments could be custom tailored to the health of the bee using modern biotechnological tools. Looking around us, the rest of agriculture is being essentially revolutionized, hopefully towards a more sustainable and productive future. And I thought maybe bringing some of these technologies to honeybees could benefit them, like I said, again, without the use of synthetic chemicals and hopefully in an environmentally friendly way.
Yeah, it is. I think that I just want to say congratulations. This is a piece of work that I think is just beautiful. It's just beautiful. The concept is very elegant. The execution was phenomenal. The results are clean. And I always try to find something, try to be a little picky here, but I couldn't find anything. So I'm very happy.
Thank you so much for saying that. We spent a lot of time on this. This wasn't a one-season thing. This was a multi-season, multi-year vetting the technology, reproducing our results. We have a supplementary materials section associated with this paper. That's about as long as 2 additional papers. That is just additional information and experimental repeats, kind of just showing the robustness of the results.
No, that's the way it's supposed to be. You only release the information when you're sure, which is that's the way science is supposed to be. But the system have their ways to push people to do things faster than they supposed to, and you know what I'm talking about. Mm-hmm. So it's not the case here. So can you guide us through what we were looking here in this figure?
So these figures are pretty large. A beekeeper looking at this first figure, it might be a bit confusing because it's pretty technical. It kind of lays a foundation for, hey, we said we have this technology that could be fed to bees to reduce deformed wing virus, and this kind of lays the technical foundation for that. So using genetic engineering technologies is what these first panels are showing us. We developed a blue-green algae, also called a cyanobacterium, that can function as an antiviral treatment for bees. So the algae has been engineered to produce these double-stranded RNAs, or dsRNAs, that correspond to small fragments of deformed wing virus. And when this panel here on the left, when the algae is mixed into food and fed to bees, they get some nutrition, but the digestion of the algae actually releases the dsRNA inside the algae, which triggers an immune response against DWV. And the fact that the dsRNA is encapsulated within the algae actually provides some additional stability to that dsRNA, which is potentially advantageous over similar technologies.
It is true. When I was working with RNAi in the past, we have a problem of delivery. And delivery was always a very hard way to use the technology. We knew for sure that when we give the molecule itself in high quantities to the organisms that we are targeting, we're able to get phenomenal results. But when we want to bring this technology to real life situations, the delivery method was always a barrier that complicates the whole system. And the way you did here to produce inside of the algae already, so that is the delivery system. I think this is brilliant because that go through the digestive system, and that was the problem for many of the organisms that we worked in before. When you put the molecule isolated, the digestive system destroyed the molecule never reached the cells or in enough quantity to trigger the response that we were looking for. And the way you did is just perfect because it goes straight where it's supposed to be, inside the cells and the digestion, and then you got the results right there. Fantastic.
Thank you.
I have a question. Do you know a little bit about the overview about using these algae as nutrition sources or something? There is any potential complication with those, or is it a normal thing that has been used in other fields?
That's a good question. So the use of blue-green algae or cyanobacteria as a nutrition source, that's been in the literature now for both humans and livestock animals for almost 40 years now. So blue-green algae or cyanobacteria, especially spirulina, is kind of the most commercially and industrially produced algae biomass in the world. It's sold as nutrition supplements for humans. There's the literature is kind of full of health modulating properties of these blue-green algae biomass. Of course, blue-green algae are pretty diverse, and there are other not so good blue-green algae in the environment too that cause these kind of toxic algae blooms. But those are just different species, whereas the ones we're working with, that we work with, are commercially produced and used for human consumption, livestock consumption, and things like that.
Yeah, that's exactly the point that I want to bring here, because my wife was looking at this, and one of the actually was my daughter that showing the first thing that came to her mind was those algae that show up in the oceans, and people have those associations with algae growing fast as a bad thing, and I'm sure people at home and beekeepers, some of them, it's going to have the same concept in their minds because they're listening to the same news. So I just want to emphasize that this is not the same species. This is a species that is being used for 40 years, apparently, in many different kind of nutritional systems out there with many different organisms. So apparently it's very safe. Cool. So you got this, you create the technology, you put the technology inside the algae, you feed to the bees don't die, apparently everything is stable. the bees consume everything. And how do you know it worked against the virus? Where should I go?
Yeah, so let's go to the next figure. And
I want you to, this is the one?
Yep.
All right.
Yeah, so, okay, so we just discussed, hey, we developed this technology. This engineered algae produces double-stranded RNAs that can induce an RNAi response in bees. And then we go on to report thorough testing of this new algae in bees that were experimentally infected with deformed wing virus, or DWV. So we grew up these strains, we formulated it into a pollen patty, and we fed it to bees. And then we injected adult bees with DWV to mimic transmission by Varroa mites. And we found that bees given the engineered algae had significantly better survival, and we reduced DWV levels by tenfold. So that panel on the left there, panel B, it's important to note that that's on a log scale.
Yes.
So we reduced DWV levels by about tenfold. So bees given the anti-DWV algae had 10 times lower virus than bees given the control.
So this is the control?
That's the control there, yep.
And this is the amount of algae that you give. The more algae you give, more response you got?
No, so actually what we have there are, we're looking at 4 different engineered algae strains.
Oh, okay.
The gray box there is a control. It's just a wild type algae that we haven't modified in any way. The yellow is a non-specific dsRNA.
Sure.
And the two green boxes there, those represent 2 different algae strains that target 2 different parts of the deformed wing virus genome. So if you look at panel A, you could see kind of a schematic of the deformed wing virus genome, and you could see where our 2 different algae strains correspond to those parts of the genome. So, if we think about the gray box as being, well, just a control, that would be as if bees received no engineered algae at all. But instead, to be scientifically sound, we gave them wild type algae that had not been modified at all,
yes.
Just to kind of rule out any sort of algae specific effects on viral replication.
Beautiful design. The algae itself, the algae with something that is not specific to the virus, and 2 different algae is with 2 parts of the genome of the virus specific against that virus. Perfect design.
Thank you. So we were able to reduce DWV by about 10 times lower than control in these engineered strains. And we also showed in panel C then upregulation of dicer, which is a gene that encodes kind of an initiating step in the RNAi pathway in bees. So we see that bees fed dsRNA, regardless of if it was specific or not to DWV, dsRNA alone upregulates the RNAi pathway. So that just kind of confirms that this reduction in viral load that we see in bees fed the engineered algae, it's likely attributable to an upregulation of the RNAi pathway in the bees themselves.
So this is a confirmation that the immune system was activated. It was not something random that we don't know. We very likely, because activated these specific genes of that specific pathway of the immune system of the honeybees, this is a kind of confirmation that you activate the right pathway and you are in control here. You know what you're doing, molecularly speaking.
That's right. And so moving on then, we also see a lifespan analysis in panel D there. And that's just showing us that bees that were fed the engineered algae, specifically the engineered algae that targets the DWV genome, those bees lived about 7 days longer than bees that were given the control.
And this is with the deformed wing virus infection. Okay, so they are infected with the same strain of the deformed wing virus. shout out to Eugene Ryabov to develop this system, because this was a breakthrough to the honeybee world, an infectious clone of the formal wing virus. Eugene, if you're listening, we love you. Okay, so bees infected with the formal wing virus, when they receive the green algae, that have the medicine inside them, they live longer. Fantastic.
They do, and that's in agreement with tenfold reduction in DWV and an upregulation of their immune system.
And this is cage studies. Am I correct?
These are all cage studies, yes.
Okay. So moving on, you're testing in larvae. This was adult bees.
That's right. So up top, what I was just describing was in adult bees. Okay. We reproduced this multiple times in adult bees, and then we showed that bees reared as larvae on jelly that we spiked with the engineered algae had improved resistance to DWV. And we decided to test this because, well, in a colony setting, it's possible that larvae could be exposed to food or exposed to bees that are feeding on the food that are provided to the colony. And we wanted to understand the effects on larvae. And we found that even small doses of the algae in the larval food, when those larvae were reared as pupae, they had significantly lower DW. Well, when they were reared as pupae and injected with virus, they had significantly lower virus levels in the engineered algae group that was targeting DWV. And we took it a bit further, and we also looked at the degree of wing deformities in those individuals that were reared as larvae on jelly containing algae, injected with DWV, and then allowed to eclose. And we found that bees that were fed the DWV targeting algae had significantly reduced wing deformities compared to the control, or bees that were reared on just regular jelly.
Wow. So you were able to quantify phenotype. This is so powerful. When we start to see results that we can quantify almost visually, is because we have very good results. Normally, in the molecular world, let me explain this to the people at home very basically. We always imagine things and measuring one or two things, and we try to create a model in our head and how things are really working. And we have those experiments that we need to have very, very different controls so we can have an idea. But at the end of the day, we have models in our mind to see if something is working or not. When you start to go through this model and start to visualize the result in the organism that we're working with, this is a phenomenal proof that you are on the right path. What they did here, they fed with the medicine the larvae. They challenge that larvae with a specific amount of the formal wing virus, and they were able to quantify less wings deformed. Is that what I'm hearing here, Vincent?
That's correct.
That's amazing, because then you have the whole story. So you have the molecular model in your head with all your controls and everything, and then you're able to see the results visually and quantify phenotype. Mwah, mamma mia. Beautiful. That excites me.
Thank you, Humberto. And you're getting me excited too. Thinking about this, it's interesting to think about projects through the lens of someone else who isn't thinking about this every day. So
yes,
that's fun.
It's beautiful. It's beautiful, and you can see results. And I think this is something good for beekeepers. Beekeepers always rely in visually signs. This is going to be one for them to. And we're going to talk about this because I have questions about, I know there is a patent. I want to know if there is any potential timeline for this coming to the market or something like that. But we're going to get there soon. But for the beekeepers at home, it's going to be easy for you. If this comes to the market, you're going to be able to see results and also be able to quantify the return of investment, ROI. So, we have a lot of supplements, we have a lot of things, and people get distracted sometimes and don't know if what they're paying and have some return. In this case, you're very likely going to see very easy. I think it's going to be very easy to see results, and you'll be comfortable to move forward with whatever product is going to come from this. And to be completely disclosed, I don't have any I don't have any financial thing. I'm just curious. I'm just a guy outside. I don't know anything about what's going on with potential companies that's going to come from this technology. Just a disclaimer, I have no idea. I'm just excited as a potential customer.
So then I suppose to summarize then, the study reports a scalable and sustainable technology that can be directly added to supplemental feed to control viruses without the use of synthetic chemicals. There's no processing involved in the production of the algae biomass to get it into feed, of course, besides, mixing with sugar and pollen substitute. Of course, there are major regulatory concerns since we're dealing with an engineered organism that need to be addressed before these applications can be fully realized and the technology deployed in the field. So it's just important to note that everything that we're looking at here has been done in the laboratory under biocontainment, and that's kind of where we're at the moment.
Maybe we should discuss this a little bit because from the beekeeping community, I think there is a lot of, pushback against genetic modifications and things like that. And I'm a defender of genetically modified when used correctly. Remember, anything in life, any tool can be used for good or for bad. In the case of genetically modified organisms, for example, one use that I hate is when pesticide companies want to make plants that are, instead of the plant to be resistant to the pest, they want to make a plant resistant to the pesticide they sell. This is ridiculous. I'm completely against that. That's a bad way to use genetically modified organism. But in this case, I think it's a fantastic way to use genetically modified organisms. It's going to reduce pesticide use. It's a very specifically oriented to a specific virus. And if we see if there is a problem with that in the future, we change. We are human beings. We have brains. We're going to realize that we have a problem, and then we fix it. Instead of pesticide companies that keep finding these loops and selling more and more, even though they know something is wrong. So I just want to poke the pesticide company a little bit. So where are we going from here? What's the next figure? Did you test this in the field?
No, we have not tested it in the field. Like I said, there's still some major regulatory considerations and risk assessments that would need to be done before this technology could be deployed in the field. So all the data were obtained in a laboratory setting.
So let me just you and me here. What did they ask? What the EPA and is the EPA or FDA also involved on that?
So I haven't really explored too much regulatory considerations just yet. The technology is still really new. We just published this paper, 2 months ago. Yeah. So now I'm just really starting to wrap my head around and beginning to understand what are some of the regulatory routes that could be taken to get something like this approved. So it's still really early, and admittedly, I'm still learning a lot about these processes. Like I said, I'm just the scientist who developed the technology in the laboratory, and now I'm trying to understand, well, what does it take really to get approval for something like this, so it could potentially see the light of day. And as I've mentioned, we're taking different routes to antiviral development of antiviral treatments, just to hedge our bets a little bit, because we don't know what type of regulatory hurdles we may face
Yes.
just yet.
On that side, I have a little bit of experience with that, but things I think got much better now since there is some breakthroughs, some companies that already push some things through, and we have a little idea about potential places to look. Because when a new technology arrives, we never really know where to look for problems. Even the EPA, just to defend the EPA a little bit, it is a new technology. We don't know. We just need to imagine potential targets and potential problems that this can come, and ask the researchers to provide some data on this regard. But a lot of those are guessing, because it is new for everybody, including the EPA. So we need to be vigilant and watch this very closely. Fingers crossed that we don't find any problems along the way. And once in the market, we still need to keep our eyes open, because that's where when it's going to be spread, and other situations might come up that we didn't imagine before. So just keep vigilant and don't be afraid to find problems. I think that's the message. Vincent, you guys patent this, so that's just for security, or there is any plan already? Something going on that you can share with us?
So no plan yet. We have some industry interest in the technology. Started talking with some companies who are interested. But of course, the goal of the ARS is to get technology into the hands of industry. So of course, there's no real monetary goal for me or for the ARS to patent something. It's really just to incentivize companies to pick up this technology and produce it and make it available to our stakeholders, to the industry. And that's really how the decision to patent the technology was arrived at.
Okay. So it's part of the job. You guys, the mission of the lab is to produce information that can be used to the public. How that's going to reach the market is companies that need to reach to you guys and try to fight to get the license from you. They need to come to you, convinced that they are the people that have the meanings and the resources and the responsibilities to develop this further, and then everybody wins. Cool. Well, if I can help in any way, whatever, I'm available to discuss. That's what I do for a living, to help companies to bring new products to the market. So I'd love to be involved. If any of these companies are there, talking with them, please hire me. I'm excited about this. That's my selling pitch here. Vincent, you mentioned natural products. Can you give a little overview about that? Since you are very smart, you shooting many different directions, because this could take a while to go and make a difference. But natural products is a completely different story. Once you identified something that has some effect, can immediately be used by beekeepers. So
that's right.
Anything you can share with us, like that to not compromise the work you're doing right now?
So natural products have been a source of drugs and the inspiration for drugs, for human drugs, for a long time. And plants, fungi, animals all produce different metabolites that help them deal with environmental stressors, help them deal with predators and herbivores. These molecules have a complexity that we simply can't just arrive at synthetically in the lab. The natural world has done a really good job of its own research and development over millions of years, finding the structures of things that do very specific jobs. And newer technologies allow us to screen large numbers of natural products for different drug activities and drug targets. And we are doing that with bees, specifically targeting viruses looking at diverse natural products, ideally trying to look at sources of natural products that aren't currently being investigated by other people. And that's about as much as I can share at the moment. We are kind of just embarking on a large screen of some natural products. And
Cool.
I'd be happy to talk with you again in the future about it.
When the time comes, yeah, I will be poking you. Don't worry about that part.
All right.
Vincent, do you have any other message to beekeepers out there, or any other projects you want to share? Otherwise, I'm going to wrap this up for us.
No, I'd say that's it for now. Thanks for your interest in the work. This is a great chat, and I'd love to revisit the podcast again if you'll have me on in the future, talk about some other work coming out of the lab.
Oh, definitely. I'll be looking for those publications. Vincent, thank you, man. Thank you not only for the great work that you do at the Bee Lab over there, the USDA, and also for your time here with us today. I really appreciate it.
It's my pleasure. Yeah, I enjoyed this. Thank you.